CRISPR-Cas9 system was discovered as a result of observations in the mid-2000s. And during this time, researchers who were studying bacterial sequences noticed that there were these repetitive palindromic sequences. So the palindromic sequences would then have these unique sequences that were flanked by these palindromic sequences. So the unique sequences were found to be representative of viral genomes that the bacteria would keep if and when it survived an infection. So a bacteria that had this CRISPR system would keep these snippets of viral genomes and flank it with these repetitive sequences if and when it survived an infection by a particular virus. And so it turns out these are adaptive immune responses from bacteria. These repeats would be processed and it would then associate with another RNA molecule before it can then bind this DNA cutting enzyme known as CRISPR associated. And so it's given this Cas. You can have different Cas, CRISPR associated enzymes, which are able to cut DNA.
So the first step is to go to NCBI Entree, and there you can search for the gene that's responsible for the disease. So in NCBI Entree, you can type in the gene that's been identified to be the cause of the pathology that you are studying. First thing then is to get the sequence of the gene, and then we need to focus on the area that we want to do the editing in. So you know the sequence of your target gene. Just knowing the sequence is not enough. You need to know the structure. So you need to know where the exons are, you need to know where the untranslated regions are, you need to know where the start codon is, you need to know if there are signal peptides, you need to know what the start site is, and so you have to look at the structure and annotate where each one is, because that's going to be important in terms of which guide RNA you ultimately select. And where do you get all the information about your transcription start site, your 5' untranslated region, the start codon, your signal peptide, the actual mature protein, your exon-exon junction, your stop codon, and your 3' untranslated region? Where are you going to get the information from? Well, it's in your National Center for Biotechnology Information, the NCBI entry that you would have searched for the gene. So just at the bottom, it will tell you where all those information are. You can go ahead and annotate it. So you take a copy of the gene, the sequences, the nucleotide sequences, and you can go ahead and mark off where each of these positions are. Because then when you look for your PAMs, your protospacer adjacent motifs, you will be able to see where those particular PAMs are. Are they in a transcription start site? Are they in an untranslated region? Are they in an exon-exon junction? Are they in a stop codon? You'll be able to see that straight away, and so that helps in determining which guide RNA to go ahead with. So the untranslated regions, for instance, if you're wanting to make a mutation in a gene, you stay away from that region because they're actually not translated. They do not code for the protein, and so it wouldn't form part of the final product. So if you mutate there, it's likely not to affect the gene in the way that you want it to be affected. And as you know, you wouldn't want to target an intron because the introns are definitely not coding regions. You wouldn't pick any guide RNA that's targeting an intron. Signal peptides also tend to guide the protein to the location in the cell that it needs to be. And so targeting those would not affect the protein per se, because they're more like the signal for where the protein should go. So you're not affecting the actual protein, but just the signal. So once again, it's less likely to lead to a mutation or an edit that you want. One way to design is to do it manually. So you just look for NGG motifs, and don't forget, because it's double-stranded, you can go ahead and look for the complementary strand. The coding strand can have NGG, but don't forget it could also have CCN, because the complementary strand would then be the NGG, right? So you can look for either NGG or CCN, because the GG complementary strand is CC and then any nucleotide. Then you want to copy 20 base pairs towards the 5' end. If you remember the structure of DNA, it's 5' to 3'. The polymerase reads it from 5' to 3'. So you want from left to right. So you're gonna look for the PAM, and then you're gonna go to the left of the PAM, also known as upstream, also known as 5'. There are multiple names for it, so you pick whichever name makes sense to you. But basically the PAM is NGG or CCN, and then you go ahead and pick the 20 base pairs that's upstream or on top of it or on the left hand side of it. But of course, if it's the CCN, then it's the other way because you're coming from the 3' or the downstream, so look for the other way. First thing then is to get the sequence of the gene, and then we need to focus on the area that we want to do the editing in. Just knowing the sequence is not enough. You need to know the structure. So you need to know where the exons are, you need to know where the untranslated regions are, you need to know where the start codon is, you need to know if there are signal peptides, you need to know what the start site is, and so you have to look at the structure and annotate where each one is, because that's going to be important in terms of which guide RNA you ultimately select. And if you are interested in joining the study group, go ahead and hit the subscribe button, and I will catch you in the next video. God bless you and take care.